Asexual reproduction is a method of plant propagation that occurs without the use of seed or fertilization. In this practical, asexual reproduction is demonstrated using 4 vegetative propagation techniques. These include natural methods such as bulb, rhizome, and stolon, as well as an artificial method using cuttings. Each propagation method allows a plant to grow from existing plant part. The propagated plants are then offset over a period of 3 weeks to study their growth and development.
The objective of this practical are to demonstrate different methods of asexual reproduction in plant and to identify suitable plant species for each propagation technique. This practical also aims to explain why asexual reproduction is preferred over sexual reproduction for the selected plant, especially due to its ability to produce plants with identical characteristics. In addition, proper vegetation propagation methods are applied to produce rapid and multiple offspring. Finally, the growth and development of the propagated plants are observed and recorded over a period of 3 weeks.
The apparatus that we use in these practicals are scissors, cutters, polybags, and bottle.
Now, let's take a look for the preparation that we did.
The preparation for these practicals begins with preparing the planting media. First, water was cutted and prepared to serve as container for planting onion bulb. Next, holes are made at the bottom of the container to allow proper drainage. The planting media is then prepared for each plant species including phyto, ginger, snake plant, and onion. After that, the plants are watered to induce root growth, and only when the top of the 1 to 2 cm of soil is dry to prevent rotting. Finally, the plant species are carefully planted in their respective media, ready for propagation process. In this practical, 4 types of plant are used to demonstrate asexual reproduction. First, ginger, which propagates through rhizome. Second, red onion, which reproduce using bulb. Third, phyto, which spread through stolon. And fourth, snake plants, which can be propagated through cutting. Each of these plants represent a different method of vegetative propagation, allowing us to observe how new plants develop without the use of seed.
Sexual reproduction in bulbs occurs through bulb formation. The fleshy scale leaves are modified into an underground stem, which stores food to support the growth of shoots and roots. The parent bulb produces the daughter bulbs or bulblets, which can be separated and planted. Each bulblets will grow into a new onion plant identical to the parent, allowing rapid and efficient propagation without seeds. Sexual reproduction is not suitable for onion for several reasons. First, it requires a longer time, as the process of seed development takes much longer compared to vegetative propagation. Second, it can reduce the size and quality of the bulb because energy that would normally go into bulb enlargement is instead used for flower and seed formation. Finally, sexual reproduction in onions relies heavily on environmental factors, such as photoperiod and temperature, making it less reliable for consistent crop production. On this slide, we can see the red onion bulbs were planted in soil with the top part facing upwards. The bulbs were watered regularly and placed in an area that receives sunlight. Over time, the bulb uses its stored nutrients to produce new shoots and roots. After 3 weeks, we can clearly see the results. The onion bulbs have grown long green shoots above the soil. This shows that asexual reproduction through bulbs is effective because the plant grows faster and does not require seeds or pollination.
Ginger grows mainly through asexual reproduction using a rhizome. A rhizome is an underground modified stem that grows horizontally below the soil surface. Each rhizome has nodes and internodes that contain buds. Under suitable conditions, these buds sprout into new shoots above the ground. At the same time, adventitious roots develop from the nodes of the rhizome to anchor the plant and absorb water and nutrients. The rhizome also stores food reserve, which support the early growth of the new plant. Because of this, each rhizome segment can grow into a new ginger plant. Sexual reproduction is not suitable for ginger because it rarely produces viable seeds, making this method unreliable. Compared to vegetative reproduction, sexual reproduction is much slower. Plants grown from seeds also show genetic variation, which can cause uneven growth and inconsistent quality. A uniform plants are preferred to ensure consistent yield and quality. Rhizome reproduction produces true-to-type plants that retain desirable traits. In addition, vegetative reproduction has a higher success rate and allows faster establishment. It also enables year-round propagation without depending on flowering or pollination. Here is the result of the 3 weeks. On the left, you can see the ginger peas plant in the soil. Over time, it absorbs moisture and nutrients from the soil. On the right, the ginger has started developing roots and small green shoots. This shows that ginger can successfully grow through vegetative propagation. The appearance of roots and shoots indicates healthy growth and confirms that the planting method was effective. With continued care, this ginger plant will keep growing and eventually produce new rhizome.
Next, snake plant. Snake plant primarily use asexual reproduction such as rhizome division and leaf propagation in water or soil. Rhizome division start from digging up parent plant, separating the rhizome near the base, and replanting it to form a new plant. In leaf propagation, the new root and shoot derive from the cut bottom of the leaf to produce new plant. In nature, it can reproduce sexually with the help of pollinators such as moths to produce seed. Then, the seed germinate under suitable conditions to form new snake plant. Sexual reproduction of snake plant requires specific conditions such as bright indirect light, constant temperature, and more than 3 years growth. However, the successful rate to flower is very low, even hit those requirements. If snake plants successfully germinate, the production of seed is inconsistent and low germination rate, while choosing asexual reproduction which have high survival rate. After 3 weeks, due to rotting, it failed to grow new root because we didn't let the cutting dry out for a few days to form callus prevent bacterial invasion. In addition, snake plant is slow grower, need at least 6 to 8 weeks to develop new roots.
Pothos reproduces through asexual reproduction using stolons, which are horizontal creeping stems that grow along the soil surface. The stolon has nodes at regular intervals, and when a node touches moist soil, adventitious roots develop from it. From the same node, a new shoot and leaves grow upward. The new plant becomes independent once the stolon dries or is separated from the parent plant, allowing it to grow on its own. Sexual reproduction is not suitable for pothos for several reasons. First, pothos rarely produces flowers, especially when grown indoors or under cultivated conditions, so pollination and fertilization usually do not occur. Second, without pollination, seeds cannot form, making sexual reproduction impractical. Finally, even if seeds were produced, growing pothos from seeds is slow and unreliable compared to vegetative propagation. In week 1, the pothos cutting had 4 shoots and 4 leaves. By week 3, it increased to 6 shoots and 6 leaves, showing active growth through vegetative reproduction without sexual reproduction.